Somewhere on a dark beach on the Atlantic coast, on the highest tide of a spring full moon, the sea begins to move in a way that looks almost wrong. Thousands of armored, dome-shaped bodies are hauling themselves out of the water and onto the sand. Ancient, helmeted things dragging long, spiked tails behind them, piling on top of one another in the moonlight to do what their ancestors have done in almost exactly this way for over 400 million years. This animal was already old when the first dinosaurs appeared. It was crawling up onto beaches like this one before trees existed, before flowers existed, before life had fully committed to living on land at all. It has survived every mass extinction the planet has ever thrown at it, including the one that wiped out the dinosaurs and the far worse one before it that nearly ended life on Earth entirely. And here is the part that should stop you. This creature, which is not a crab, does not have blood like yours, and carries roughly 10 eyes, including some on its tail, is very likely the reason you are alive. Almost every vaccine you have ever received, every injectable drug, was proven safe using the pale blue blood inside its body, including the COVID-19 vaccines. This is the horseshoe crab, and almost nothing you assume about it is true. It isn't a crab, its tail isn't a weapon, it has no jaw, and it chews its food by walking. Its blood is blue, and for one strange chemical reason, it may be worth more, drop for drop, than almost any liquid on Earth. And right now, in the year 2024 and 2025, a single quiet decision made in a book of pharmaceutical rules may be about to change this animal's fate forever. So what exactly is this thing if it isn't a crab? How did a beach animal's blood end up guarding the entire modern drug supply? Why does a bird that flies from the bottom of the world to the top of it live or die by this creature's eggs? And why are scientists now arguing that this humble survivor might secretly be a spider? I'm David, and you're watching Phoenix. Join me as we uncover an animal that outlived the dinosaurs, whose blood built modern medicine, and whose true identity is quietly rewriting the family tree of life on land. Let's clear up the biggest lie first, the one hiding in the name itself. The horseshoe crab is not a crab. It is not even a crustacean. It belongs to a completely different branch of life called the chelicerates, the same group that contains spiders, scorpions, mites, and the long-extinct sea scorpions. Its closest living relatives are not crabs scuttling on the seafloor. They are the arachnids. In a very real sense, what you are looking at on that beach is closer to a spider wearing a helmet than to anything you'd find on a dinner plate. The confusion goes right back to the beginning. When Carl Linnaeus, the father of scientific naming, formally described the American species in 1758, he made a small joke that stuck. He placed it in a group he called monoculus, meaning one-eyed, and named the species Polyphemus after Polyphemus, the one-eyed cyclops of Greek myth. It's a double joke because this animal is the opposite of one-eyed. It has around 10 eyes. Linnaeus knew that and named it after a one-eyed giant anyway. The body itself is essentially a walking suit of armor in 3 parts. At the front is the large horseshoe-shaped shell that gives the animal its name. Behind that sits the rear section, edged with movable spines, and trailing from the back is the long, pointed tail, the telson, the single most misunderstood part of the entire animal. Because here is what almost everyone gets wrong. That tail is not a stinger. It is not a weapon. It cannot hurt you at all. Its real job is to be a lever. When a wave flips the animal onto its back, which happens constantly on a crowded spawning beach, it arches its body, jams the tail into the sand, and levers itself right side up again. A horseshoe crab that loses its tail is a horseshoe crab far more likely to die, stranded and helpless in the sun. This is why the volunteers who patrol these beaches have one iron rule. If you find one flipped over, turn it by the shell. Never, ever by the tail. Now flip the animal over gently, and its strangeness only deepens. Because a horseshoe crab has no jaw, none. It has no mandible, nothing to bite with. So, how does it eat? It chews with its legs. The bases of its walking legs are covered in hard, spiny, grinding surfaces called gnathobases. As the animal walks, those leg bases crush the food between them. Worms, small clams, mollusks, whatever it can find, grinding it up and pushing it forward toward the mouth, which sits in the very center of the body, surrounded by the legs. Think about what this means. For a horseshoe crab, eating and walking are mechanically the same act. To chew its dinner, it essentially has to keep moving. A horseshoe crab standing perfectly still is a horseshoe crab that cannot eat. Then there are the eyes. We tend to picture 2 eyes on an animal because that's what we have. This creature has around 9 or 10 light-sensing structures scattered across its body. 2 large compound eyes on the sides of the shell, a pair of median eyes, several smaller and simpler eyes, light-sensing organs near the mouth underneath, and, this is the strange one, photoreceptors running along the tail itself. Sit with that for a moment. This animal has light sensors on its tail, and they are not decorative. They feed into the creature's internal body clock, its sense of day and night, which means that even when the horseshoe crab has buried its head in the sand, its tail can still see the difference between daylight and darkness and keep its rhythms in time. It is, in the most literal sense, an animal that senses the world partly through its tail, and its eyes did something no other animal's eyes have done. But we'll come back to that, because that story ends with a Nobel Prize. First, the blood. Cut a horseshoe crab, and what wells up is not red. It is blue, a pale, milky, almost otherworldly blue. The reason is chemistry. Your blood carries oxygen using hemoglobin, a protein built around iron, and iron turns red. The horseshoe crab uses a completely different molecule called hemocyanin, built around copper instead of iron, and copper, when it grabs oxygen, turns blue. This blood doesn't even travel the way yours does. Instead of being sealed inside tight vessels, it partly bathes the animal's organs directly in open spaces inside the body. But the color is not what makes this blood one of the most valuable liquids on the planet. What makes it priceless is a single type of cell floating inside it, and the astonishing thing that cell does when it meets bacteria. And that is where the horseshoe crab stops being a curiosity and starts becoming the quiet foundation of modern medicine. Now flip the animal over gently and its strangeness only deepens, because a horseshoe crab has no jaw, none. It has no mandible, nothing to bite with. So how does it eat? It chews with its legs. The bases of its walking legs are covered in hard, spiny, grinding surfaces called gnathobases. As the animal walks, those leg bases crush the food between them. Worms, small clams, mollusks, whatever it can find, grinding it up and pushing it forward toward the mouth, which sits in the very center of the body, surrounded by the legs. In 1956, a pathologist named Frederick Bang was studying horseshoe crabs when he noticed something that made no sense. When he infected them with certain marine bacteria, their blue blood didn't just react, it clotted massively. The blood turned almost to gel. Something inside it was detecting the bacteria and locking down around them, trapping them in a solid clot. Working with a hematologist named Jack Levin through the 1960s, Bang tracked down exactly where this reaction came from. The blood of a horseshoe crab contains essentially one dominant type of immune cell, the amebocyte, and it is these cells that sense the invader and clot around it. Bang and Levin found they could burst these cells open, take the fluid inside, and bottle that reaction. They called it limulus amebocyte lysate, LAL for short, and LAL turned out to be the most sensitive detector of bacterial contamination that science had ever found. To understand why that matters so enormously, you need to understand a killer called endotoxin. Endotoxin is a molecule found on the outer wall of certain bacteria. And if even a tiny trace of it gets into your bloodstream, say, in a drug injected into your arm, it can trigger fever, plummeting blood pressure, shock, and death, which creates a colossal problem for medicine. Every single injectable drug, every vaccine, every implanted medical device, anything that goes inside the human body has to be proven completely free of endotoxin before it can be used. For a long time, the only way to test for it was slow, clumsy, and grim. Inject a sample into a rabbit and wait to see if it developed a fever. Then came LAL. Drop a little horseshoe crab lysate into a drug sample, and if there is even a picogram of endotoxin present, that's a trillionth of a gram per milliliter, the lysate gels. Instantly, cleanly, and with a sensitivity nothing else could match for decades. In other words, a 400-million-year-old immune defense turned out to be perfectly tuned to detect the one molecule modern medicine most needs to screen out. The chemistry behind it is beautiful in its precision. The endotoxin trips a chain reaction inside the blood, one protein activating the next, like a line of falling dominoes. Factor C wakes up Factor B, which wakes up a clotting enzyme, which converts a protein called coagulagen into coagulant, and the coagulant knits itself together into that visible gel. In the living animal, this same cascade physically traps bacteria at the site of a wound and seals its shell against infection in filthy, bacteria-rich water. It is a self-healing antiseptic bandage, evolved over hundreds of millions of years. We simply took that ancient defense, put it in a test tube, and pointed it at our own medicines. So the next time you hear that horseshoe crab blood safety tested the COVID-19 vaccines, understand that this is not an exaggeration. For decades, essentially, the entire global supply of injectable medicine has passed through a safety check that depends on the blood of this one ancient beach animal. All of which raises an uncomfortable question. If we need this blood so badly, where does it come from? The answer is, from the animals themselves, taken from the wild. Every year in the United States, somewhere in the region of 400,000 to 500,000 horseshoe crabs, and by some accounts more, are collected from the beaches, brought to laboratories, and bled. A needle goes into the area near the heart, and up to around 30% of the animal's blood is drawn out. Then most of them are trucked back and returned alive to the sea. And this is where I have to be honest with you, because the honesty is the whole point. You will often hear the processed lysate quoted at something like $15,000 a quart. That figure gets repeated so constantly that it has become a kind of folklore, and the exact number should be treated with caution. It may well be dated or inflated, but the underlying truth it points to is real. Drop for drop, this is one of the most valuable liquids humans harvest from any animal on Earth. The more serious dispute is over how many crabs the process kills, and this is genuinely unresolved. Estimates tied to the industry have historically put the death rate low, around 3 to 8%, but independent studies have suggested the real figure could be far higher. Somewhere between 10 and 30% of the animals bled. The two sides use different methods and different time frames, and the true number remains contested. On this, the honest position is not to pick the convenient figure, but to say plainly, we do not fully know and the range is wide. But here's the part that the survival statistics quietly hide. Even the crabs that live are not necessarily fine. Research through the 2000s and 2000s has found that bled females spawn less. They move differently. Their internal rhythms, the very tidal clocks that tell them when to come ashore and breed, can be disrupted for weeks after they're released. So an animal counted as a survivor may still be, in reproductive terms, effectively lost for a season. The real cost of the blood is larger than any mortality number alone can capture, and that is a fact worth stating carefully rather than dramatizing. So for decades we've been caught in a bind. We need the blood. Taking it harms the animal, and you cannot simply farm horseshoe crabs to solve the problem. Attempts to raise them at commercial scale for bleeding have largely failed, which means our entire drug safety system has been tied directly to the health of wild populations. But in the last few years, science has finally offered a way out. It's called recombinant factor C, or RFC. Remember factor C, the very first protein in that clotting chain reaction, the one that actually detects the endotoxin. Scientists worked out how to clone it, to manufacture that single sensing protein synthetically in the lab without any crab involved at all. And it works. It detects endotoxin with specificity equal to or better than the crab blood version, while taking the animal out of the supply chain completely. For years, though, adoption stalled on what sounds like the most boring obstacle imaginable: paperwork. The synthetic version worked, but in 2020, the United States Pharmacopeia, the body that sets the official rulebook for drug testing, declined to give RFC the same standing as the traditional crab-based test, keeping it as a mere alternative that required extra hoops to use. Europe moved faster to accept it. America dragged. And then came the turning point. In 2024, the US Pharmacopeia finally published a new chapter formally recognizing these recombinant reagents for endotoxin testing, taking effect across 2024 and 2025. It is, on paper, an almost absurdly dry document, a bureaucratic paragraph in a technical manual, but its consequences may be enormous. A single regulatory paragraph may end 60 years of bleeding, and it may be the biggest thing that ever happened for this animal's survival. Think about the strangeness of that. Not a heroic rescue mission, not a nature reserve, not a breeding program. A change of wording in a book of pharmaceutical standards may be what finally lets the drug industry move off crab blood at scale and takes the needle out of this creature for good. Sometimes conservation doesn't look like a wildlife documentary. Sometimes it looks like a committee updating a chapter number. But the horseshoe crab's importance doesn't end with human medicine. Long before we ever discovered its blood, this animal was holding up an entire ecosystem, and the story of how connects a beach in Delaware to the very tip of South America and the high Arctic all at once. It comes down to eggs. When those thousands of crabs haul ashore on the moonlit spring tides of May and June in Delaware Bay, the females bury clutches of eggs in the sand. A single female may lay on the order of 88,000 eggs across a season. Most stay safely buried. But as wave after wave of later nesting females dig into the same beach, they churn some of those eggs up to the surface. And those surface eggs, the small overflow fraction, the ones that were never going to hatch anyway, become one of the most important food sources on the entire Atlantic coast. Enter a small bird called the Rufa red knot. This bird undertakes one of the longest migrations of any animal alive, from Tierra del Fuego at the southern tip of South America, all the way to its breeding grounds in the Arctic. And it makes that impossible journey with a single, precisely timed refueling stop. It arrives in Delaware Bay exactly when the horseshoe crabs are spawning, and it gorges on their eggs so ferociously that a red knot can nearly double its body weight in a matter of days, loading up the fuel it needs to finish the flight to the Arctic and breed. This is a synchronization of staggering precision, the moon-timed spawn of an ancient sea creature, the tides, and the arrival of a bird that has flown from the bottom of the planet. All three have to align within a narrow window each spring. It is astronomy and biology locked together across a hemisphere, and in the 1990s, we nearly broke it. When horseshoe crabs were over-harvested and their egg numbers crashed, the red knots arrived to find the table nearly bare. The birds couldn't refuel fast enough, and the consequences showed up thousands of miles away. The Rufa red knot population fell dramatically, by some counts around 75%. and in 2014, the bird was formally listed as threatened under the U.S. Endangered Species Act. A bird that crosses an entire hemisphere was pushed toward the edge because of how many crabs were taken from one bay. And the birds need more eggs than you might guess. Research suggests they require extraordinarily high densities of surface eggs, thousands per square meter, to fatten quickly enough. Below a certain threshold, they simply can't gain weight fast enough to survive the journey. It isn't a gentle slope, it's a cliff edge. And the red knot is only the most famous of the dependents. Ruddy turnstones, sanderlings, sandpipers, and more all rely on the same pulse of eggs, while sea turtles, sharks, and fish feed on the crabs themselves. Pull out the horseshoe crab, and a whole web of life along the coast begins to unravel. It's worth remembering how carelessly we treated this animal before we understood what it was worth. For a stretch of history running from the late 1800s into the mid-1900s, horseshoe crabs were harvested along the Atlantic coast, not for medicine, not even for bait, but to be ground up into fertilizer and livestock feed. And the scale was almost incomprehensible. In the early 20th century, the Delaware Bay harvest alone reportedly reached something like 1.5 to 2 million crabs a year. Millions hauled off the beaches and turned into fertilizer year after year, a slaughter that permanently lowered the baseline population from which the species has never fully recovered. As fertilizer fell out of fashion, a new demand took over: bait. Horseshoe crabs became the preferred bait for catching American eel and whelk, and that, alongside biomedical bleeding, remains the main pressure on them today. Since 1998, a body called the Atlantic States Marine Fisheries Commission has managed the American fishery with harvest quotas, and Delaware Bay in particular now runs an adaptive system that explicitly ties how many crabs can be taken to how the red knot population is doing. There have been clever fixes too. Bait bags and mesh designs that cut the number of crabs a fisherman needs per trap by half or more. But here is the part of the conservation story that too often gets ignored, because the whole blue blood saga is so American. The most urgent crisis is not in the United States at all. It's in Asia. There are 4 living species of horseshoe crab in the world, and while the American one is officially classed as vulnerable, the Asian tri-spine horseshoe crab is doing far worse. Assessed as endangered, with steep declines across China, Japan, and Southeast Asia. Coastal development, harvesting for food and medicine, and the destruction of the mangrove forests they breed in have driven Asian horseshoe crabs toward local extinction in many places where they were once abundant. In parts of Asia, harvest for food markets is poorly regulated and barely recorded, so the true scale of the loss is genuinely unknown. The famous story is the blood. The urgent story is Asia. Earlier, I promised to come back to those eyes. Here's why they matter far beyond the animal itself. The large compound eye on the side of a horseshoe crab's shell has unusually big, unusually accessible nerve fibers running out of it. So large that scientists could actually record the electrical signals from individual cells, something almost impossible in most animals. That made this humble creature the perfect window into how vision works at all. In the mid-20th century, a scientist named Haldon Keffer Hartline used the horseshoe crab's eye to discover a fundamental principle of how eyes, all eyes, including yours, sharpen what they see. He found that when one light-sensing cell fires, it actively suppresses its neighbors. This effect, called lateral inhibition, is how a visual system pulls edges and contrast out of a blurry world. It is one of the foundation stones of modern neuroscience. And in 1967, it earned Hartline a share of the Nobel Prize in Physiology or Medicine. An animal that chews with its legs and can't tell you what a jaw is helped us understand how the human brain sees. And the eyes hold one more astonishment. This animal's brain actively retunes its own eyes between day and night. Through nerve signals sent from the brain out to the eye, it can crank up the eye's sensitivity after dark by around 1,000,000-fold, a factor of 1,000,000. It is one of the clearest examples known in all of biology of a brain deliberately adjusting the gain on its own sense organs, turning the sensitivity up at night the way you might turn up the volume on a radio. And feeding into that same day-night clock, as we saw, are the light sensors on its tail. Then there is its immune system, or rather its lack of one, in the sense we usually mean. Horseshoe crabs have no antibodies, no adaptive immune system that learns and remembers specific invaders the way yours does. They defend themselves entirely with raw, ancient chemistry, the clotting cascade and a set of bacteria-killing molecules. And yet they thrive in warm, filthy, bacteria-laden water that would overwhelm many animals. It's precisely because their defense is chemical and automatic rather than learned that we were able to bottle it. Their blood reacts to bacteria as reliably as a chemical test because that is essentially what it evolved to be. So how does an animal like this last 445 million years? Partly through a body that is almost comically hard to kill. Keep its book gills, the stacked page-like flaps it breathes through, even slightly moist, and a horseshoe crab can survive out of water for many hours, sometimes days, which is exactly how it endures being stranded high on a beach when the spawning tide rolls back. In the cold northern part of its range, it buries itself in the winter sediment and drops its metabolism into a near-dormant crawl. It can tolerate huge swings in temperature and saltiness. One of the Asian species lives in the brackish, ever-changing water of mangrove estuaries. It can go for something like a year without eating, and it can lose a large fraction of its blood and simply regenerate it, which is, whether we like it or not, the biological reason the bleed and release industry is even possible. It can regrow lost limbs. It can seal wounds in its shell using the same clotting chemistry we harvest for medicine, an antiseptic bandage built into its own body, and in a detail that delights everyone who learns it, both the young and the adults can swim upside down using those book gills as paddles. A horseshoe crab gliding along inverted through the water is not a mistake or a dying animal. It's a genuine way this creature gets around. Its life story is slow and strange from the very start. The young hatch as what are charmingly called trilobite larvae because they look uncannily like the trilobites that shared the ancient seas. And at first, they don't even have a working tail. From there, growth means molting, shedding the entire shell and growing a bigger one, again and again, around 16 or 17 times over roughly 9 to 11 years before the animal is finally old enough to breed. It may then live 20 years or more, and because it sheds its whole shell each time, it leaves behind no growth rings, no record of its age, which, as we'll see, is part of why we still can't say for certain how long these animals really live. Even its mating carries a twist. A male grips onto a female with specially modified hook-shaped front legs and is towed up the beach as she lays. But clustered around them are other males, satellite males, releasing sperm of their own. And genetic paternity studies have shown these hangers-on father a real and substantial share of the offspring. On these crowded beaches, where males heavily outnumber females, the tidy picture of one pair breeding quietly is a fiction. It's a scramble. Now for the deep past, and for a myth that needs correcting. You will constantly hear the horseshoe crab called a living fossil, a phrase coined by Darwin himself, as though it were some creature frozen in time, evolution's forgotten leftover. That framing is misleading, and the truth is far more interesting. The lineage really is astonishingly old. The oldest known body fossil, a creature called Lunataspis aurora, described in 2008 from rocks in Manitoba, Canada, is roughly 445 million years old. And it already had the recognizable horseshoe crab shape, which pushed the origin of the whole group back more than 100 million years earlier than scientists had thought. And there are fossils that capture these animals in heartbreaking, cinematic detail. From the famous Solnhofen limestone of Germany, around 150 million years old, one specimen preserves a 9.7-meter trackway, the animal's final walk. It had blundered across the floor of an oxygen-starved lagoon, staggering forward until it died at the end of its own footprints. Both the tracks and the dead crab that made them fossilized together. Paleontologists call it a death march, and you can still see, frozen in stone, the exact path of an animal's last moments from 150 million years ago. But living fossil gets the biology exactly backwards. Horseshoe crabs have never stopped evolving. Their genomes, their immune chemistry, their physiology have all continued to change. What has stayed stable is mainly the external body plan. And that's not a sign of frozen evolution, but of a design so good it simply hasn't needed to change. In fact, the deep fossil record shows their extinct relatives were wildly diverse. Ancient members of this group lived in freshwater and coal swamps, not just the sea. Some Triassic forms had grotesque, elongated, wing-like spines sweeping off their heads. The 4 survivors we have today are a small, narrow marine remnant of what was once a sprawling and experimental family of shapes. The single unchanging horseshoe is the exception in their history, not the rule. And now the real bombshell, the one that could reshape a corner of the tree of life. For as long as anyone can remember, horseshoe crabs have been treated as a group sitting beside the arachnids, the spiders and scorpions. But in recent years, repeated genetic analyses have recovered a startling result. The horseshoe crabs may sit inside the arachnids, not next to them, among them. If that's right, then either arachnid isn't a real natural group as we've always drawn it, or the ancestors of spiders and scorpions crawled out of the sea and onto land more than once. Either conclusion is genuinely destabilizing for how we understand the evolution of life on land. A humble armored animal that most people mistake for a crab may end up forcing biologists to redraw the spider family tree. And I want to be careful here, because this is exactly the kind of claim that gets oversold. It is not settled. It is an active, unresolved debate, but the fact that it's even on the table tells you this simple beach creature still has the power to overturn textbooks. Part of what makes it so resilient may be hidden in its genome, which shows evidence of something dramatic. Multiple rounds of whole genome duplication, where the entire genetic library was copied over, possibly up to 3 times in this lineage. That's the same kind of event seen in the ancestry of vertebrates like us, and it leaves an animal with many spare copies of its genes to tinker with, a possible source of the physiological toughness that has carried it through 4 mass extinctions. For all its gentleness, and this is an utterly harmless animal to handle, there is one genuinely dangerous fact hiding among the 4 species. In South and Southeast Asia, people sometimes eat horseshoe crabs, and one species in particular, the mangrove-dwelling Carcinoscorpius rotundicauda, can be lethal to eat. That's because it can accumulate tetrodotoxin in its body, the very same deadly poison found in pufferfish, the one that makes fugu such a notoriously dangerous delicacy. There are documented cases in the medical literature of people in the region dying after eating these animals, and the twist is almost too strange to believe. Recent research suggests the toxin doesn't even come from the crab itself, but from the bacteria and prey it picks up from its environment. So you can be killed by pufferfish poison, carried by an animal that is not a pufferfish, that is not even a fish, that is not even a crab, but is, in truth, a distant relative of spiders. It's a chain of the unexpected, almost custom-built to catch people off guard. For an animal this famous, this studied, this important to medicine, it is humbling how much about the horseshoe crab remains genuinely unknown. And in science, what we don't know is itself worth stating clearly rather than papering over. We still don't agree on the true death rate from biomedical bleeding. The estimates, as we saw, run from around 4% to over 30%, and the honest answer is that it's unresolved. We don't fully understand how adults navigate back to specific beaches to spawn, whether they follow chemical cues, the tides, the stars, or perhaps even the Earth's magnetic field, which has been suggested but never firmly proven. We don't really know where the juveniles and the deeper offshore adults spend much of their lives. A large part of this animal's existence happens out of easy human sight, and telemetry studies are only now beginning to map it. Because molting erases any record of age, we can't say with confidence how long they actually live. 20 years is an estimate, not a measured ceiling. We can't yet fully explain why the body plan has stayed so stable for so long, while the genome duplicated and the immune chemistry diversified. And, as we've seen, we haven't even settled where this animal truly belongs on the tree of life. Every one of those gaps is an active area of research right now. This is not a solved animal. It is a creature we have leaned on for 60 years of medicine and 400 million years of ecology, and still, in many ways, do not understand. So step back, finally, and look at the whole animal. Here is a creature that is not a crab, defends itself without antibodies, chews with its legs, senses daylight with its tail, and carries pale blue copper-based blood that has quietly guarded the safety of nearly every injectable medicine of the modern age. It survived whatever ended the dinosaurs, and the far greater extinction before that. It taught us how our own eyes see. It feeds a bird that crosses a hemisphere. And it may yet force us to rewrite the story of how life first conquered the land. And it stands, right now, at a genuine crossroads. On one side, a slow-breeding animal under real pressure, over-harvested for a century, still bled for its blood, and in Asia sliding toward the edge. On the other, a quiet piece of good news, a synthetic version of its blood chemistry that finally works, and a single change in a book of rules in 2024 that may, at last, let us set this ancient creature down and stop taking from it. For 445 million years, the horseshoe crab asked nothing of us. It's a strange kind of privilege that, for once, we may be about to give it something back. Its blood, left where it belongs. The next time you're near an Atlantic beach on a full moon spring tide in early summer and you see those armored shapes hauling out of the dark water exactly as they have since before the dinosaurs, remember what you're really looking at. Not a crab, not a relic, not a monster. A living bridge back to the deep past, one that has outlasted almost everything, and that has, without ever knowing it, kept you alive. If you made it all the way to the end with me, I'd love to know which fact hit you hardest. The blood that tests our vaccines, the bird that lives or dies on its eggs, the Nobel Prize hiding in its eye, or the idea that this crab might secretly be a spider. Drop it in the comments below, and I'll see you in the next one.